Hey guys, um, again, I apologize again for not being able to attend synchronously to our today's review session. I do have an emergency. My car broke down, so I will have to take it to the mechanical. But anyway, I don't think today's review session will be more important than what we are going to have tomorrow and on Monday. But I will still record this, and whoever wants to watch, I think might be useful. Of course, I will upload everything online after this meeting is over, like Word documents. Actually, Word documents are already uploaded, all PowerPoints in case you prefer learning from your computer. So I will share my screen right now and start with review session for units 8, 9, and 10. It's a long presentation. I am aiming to finish in an hour and a half. So we will see. Anyway, starting with review session for the unit 8. So about photosynthesis, you generally all know what photosynthesis is. If you ever took any plant-related class, then you heard about photosynthesis. But I am going to be very short, and I will briefly mention what photosynthesis is. But what I would like you to memorize is the equation for photosynthesis, which is on the bottom of this slide. Again, photosynthesis can be oxygenic. We'll talk about anoxygenic later on, but generally oxygenic photosynthesis involves conversion of carbon dioxide and water into complex organic molecules such as carbohydrates, and we have release of oxygen. Plants are making oxygen for our planet, but this is microbial class, and there are microbes that are also performing oxygenic photosynthesis. Generally, photosynthesis is split into light and dark reactions. And in light reactions, water is split using light into oxygen, protons, and electrons. And in dark reactions, protons and electrons that are made in light reactions are used to reduce fixed carbon dioxide to create some carbohydrate. And this is overall reaction for photosynthesis. And when you get, I would like you to be able to recognize this reaction once when you see it. So you don't have to memorize to know to write, but once when you see, you need to know what this equation represents. By the sign for delta G, you can conclude that this is endergonic reaction, which means photosynthesis is extremely energetically expensive process. Full principle of photosynthesis is nothing else but electron transport chain, ETC, with slight modifications. So pretty much as we have oxidative phosphorylation, which is electron transport chain, photosynthesis is photophosphorylation, which is also electron transport chain. So all purpose of any electron transport chain, including photosynthesis, is to have electrons going from one to another electron carrier, which will create proton motive force. So electrons are jumping from complex to complex, protons are pumped across the membrane, and that pumping of proton is creating a gradient, which is proton motive force, later on used for biosynthesis. Again, various biochemical processes will result in creating reduction equivalents, NAD, NADPH, FADH, and these NADPHs are used in oxidoreduction reactions. So photosynthesis is, I would say, similar like oxidative phosphorylation in terms of products, not completely same though, but the purpose of it is similar. So what's happening in light reactions? So in light reactions, in photosynthesis, you need to know that water is oxidized and oxygen is formed. And of course, it requires light. Well, the light that is used in light reactions is used for electron to be excited. So when photon, a light, so light or photon, so photon of light, is hitting the electron, electron gains an energy, so electron moves from basic to excited state. So that photon of light is hitting the electron in specific reaction center in photosynthesis, which is photosystem 2. So there are various photosystems, and different bacterial species have different photosystems. Plants, for example, have photosystems 1 and 2. You will see later on some bacteria have different photosystems. But the point is that in reaction center, this reaction is occurring. So once an electron is in excited state, the next is to have this electron moving through various carriers. And again, this is typical electron transport chain. So carriers and electron acceptors, here actually carriers, not electron acceptors, are plastoquinone and cytochromes. Again, you are familiar with quinone pool and cytochromes. And from there, electron goes to next reaction center, which is photosystem 1 in case of cyanobacteria. This is example of cyanobacteria. So in photosystem 1, you have ferredoxin and plastocyanin. So plastocyanin does not exist in oxidative phosphorylation. This is specific for photosynthetic organisms, but principle is the same. Now, at photosystem 1, you are getting a reduction of NAD plus to NADPH. So this is creation of reduction equivalents, and during these electron transfers, proton motive force is formed. So you do have some, so in all these electron transfers across reaction centers and carriers, you have building of reduction equivalents that will be used for further synthesis. And you have creation of proton motive force, which will also be used later. At the end, you again see formation of ATP. So basically, all purpose of light reaction is to create reduction equivalent ATPs and proton motive force and energy for further biosynthesis. And this is called photophosphorylation. So when you have typical electron transport chain that we talked about earlier, when bacteria oxidizes, like let's say organic molecule, that's called oxidative phosphorylation. This is called photophosphorylation because light is used to bring electron from lower to higher energetic state. When we talk about photosynthesis, what we talked about a few seconds ago was oxygenic photosynthesis, meaning that one of the products is oxygen. And that's done by cyanobacteria. There are other types of photosynthesis that do not involve creation of oxygen. Even though principle is similar, the final product is not O2, it can be sulfur, for example, S2. But let's go step by step. That's why it's called anoxygenic, no oxygen. So one type of bacteria that is performing anoxygenic photosynthesis are green sulfur bacteria. And the name tells you what is the product. Because it's sulfur bacteria, you can guess that they are producing sulfur. So instead of having a water oxidized, which we see in oxygenic photosynthesis, a sulfur is released because hydrogen sulfide is oxidized. So instead of H2O, you have H2S oxidized. And all remains similar. Electrons are moving through various carriers, again, quinone pool and cytochromes. And finally, electrons are coming to the reaction center, P840, which is nothing else but photosystem. It's analog to photosystems that you heard about in oxygenic photosynthesis. So reaction center. At reaction center, what's happening is electron excitation by light. So electron goes from lower to higher energy state and moves later on through various iron-sulfur complexes. Electron ends up at ferredoxin. At ferredoxin, you have an enzyme, ferredoxin reductase, that will complete oxidation of ferredoxin, but in the same time, you will get reduced NAD to NADH. Again, so this is oxidoreduction reaction that involves ferredoxin oxidoreductase. As the name tells you, ferredoxin is oxidized, NAD is reduced to NADH, and now you have creation of that reduction equivalent. This is a light phase of photosynthesis, creation of reduction equivalents, and proton motive force that will be used later for biosynthesis. The other type of bacteria that is performing anoxygenic photosynthesis are purple sulfur bacteria. And again, as the name says, you have creation of sulfur at the end. Principle is similar like to in green sulfur bacteria. What is different are photosystems, reaction centers, and carriers of electrons are slightly different. One of characteristic processes is cyclic photophosphorylation. So let's go step by step. You again have sulfur oxidation, hydrogen sulfide oxidation, and you get released sulfur. Electrons from that oxidation will reach a reaction center at photosystem 870. Note, previous one was 840. At the reaction center, electrons get excited by light. That's what we need to have. That's where light comes into the game. Now, when electron is ready to go, it starts traveling from one to another carrier. Quinone pools, BCH1, all different carriers. From those carriers, electrons end up at cytochromes, BC1, and then from BC1 travel to C2. So principle is pretty much the same. The only thing that is different is that electrons from cytochrome C2 now go back to photosystem 870, so the cycle can continue. So this is called cyclic photophosphorylation, which is typical for these purple sulfur bacteria. Okay. So now I will give a mini summary, which I titled here main categorization of phototrophs. Phototrophs, organisms that use light to generate energy for metabolic processes. So just a mini summary. Oxygenic phototrophs or cyanobacteria. In this case, we see water oxidized to oxygen and electrons used in photosystems 2 and 1, which are electron transport chains. You have anoxygenic, no oxygen phototrophs, which are green and purple sulfur bacteria, and they have hydrogen sulfide oxidized to sulfur, and nitrates and metals can be also oxidized, so not just sulfur. And electrons are used in different photosystems, which are also electron transport chains, where 870 photosystem has cyclic photophosphorylation, traveling electrons from cytochrome back to the reaction center. The third phototrophs are phototrophs that are associated with the pump bacteria rhodopsin. And we mentioned this a long time ago, I remember that. And light is used here in these organisms to turn on proton pumps. And the rhodopsin is proton pump. So thanks to rhodopsin, we have creation of proton motive force. But light is helping creation of this proton motive force. So light is turning on rhodopsin. Rhodopsin pumps proton. Rhodopsin is associated with ATP synthesis in the membrane, and you get ADP plus PI, result ATP. So basically, you have a pump in the membrane, light turns on that pump, and pump starts creating proton motive force, and also there is connection with ATP synthesis. So slightly different use of light, but it's also phototroph because light is needed to turn on proton pump. So this was all what we talked about so far are reactions that are happening and that are light dependent. Purpose of these reactions is to create reduction equivalents and energies for next phase of photosynthesis, which is dark reactions. As the name says, there is no need for light in these reactions. And here are some important components of this. So what is used in these reactions are ATP and reduction equivalents generated in light reaction. These molecules are used to fix carbon dioxide, and from that fixed carbon dioxide, bacterium is synthesizing carbohydrates, fuels for themselves. Here is important to know what are ways of carbon dioxide fixation. So there is a Calvin cycle and there is reverse TCA cycle. Probably more famous is Calvin cycle, but we will go through both of them separately. So Calvin cycle. So fixing carbon dioxide in light dependent, in dark reactions of photosynthesis. So when molecule carbon dioxide comes from the atmosphere in bacterial cell, it is going to combine with 5-carbon compound called RUBP, or ribuloso 1,5-bisphosphate. What is the result of this condensation is 6-carbon compound, so 1 carbon dioxide plus 5 carbon compounds, and this is 6-carbon compound which is the start of dark phase or Calvin cycle. This 6-carbon compound is now split into 2 3-carbon compound, which is called 3-phosphoglyceric acid, or shortly 3PG. This reaction of splitting is catalyzed by Rubisco enzyme. This is famous enzyme that you need to know very well. And I believe you all heard about Rubisco. So now 3PG, there are 2 3PG molecules that are converted into simple sugars, glyceraldehyde 3-phosphate, G3P, and this reaction spans ATP and NADPH that is made in light reactions. So everything energy related that was made in light reaction is spent in dark reactions to create sugars. G3P, I believe you already know, this is typical central carbon metabolism product. So one of G3P molecules is used to create glucose, a fuel, where the other portion is recycled to regenerate 5-carbon RUBP. Why do we need RUBP, or ribuloso phosphate, 1.5 bisphosphate recycled, is to be so we need that 5-carbon compound that will be able to capture another carbon dioxide so all cycle can operate. So in general, Calvin cycle can be divided into 3 main categories, which is carbon fixation, step 1 to 3 here, then reduction, steps 4 and 5. Sorry, my bad, steps 4 and 5 until you get glucose, and regeneration, which is step 6. Now, reverse, yeah, I forgot to mention, Calvin cycle is extremely energetically expensive. So this is important to know for general knowledge. Calvin cycle spends a lot of ATPs and NADPHs. So it's a process that really costs cell a lot. So that's not energy favorable process. Reverse TCA cycle is another way of carbon fixation that we see in microbes. So here are steps. So molecule of carbon dioxide from the atmosphere now combines with succinyl coenzyme A from TCA cycle. And this reaction is catalyzed by ferredoxin-linked enzymes. These enzymes from reaction centers of green and purple sulfur bacteria are creating alpha-ketoglutarate. So basically what you have is combination of succinyl coenzyme A and carbon dioxide, which is, this reaction is catalyzed with ferredoxin-linked enzymes from reaction centers, and you get alpha-ketoglutarate. Of course, ferredoxin goes from reduced to oxidized state. This is also oxidoreduction process. Alpha-ketoglutarate is TCA cycle component, and now alpha-ketoglutarate is reacting with carbon dioxide, and this reaction is catalyzed by carboxylase. It is adding carbon dioxide, so it's carboxylase. So TCA cycle goes in reverse. The result is isocitrate, and all these reactions are now spending reduction equivalents from light phase. So in regular TCA cycle, what you would have is basically you would have creation of reduction equivalents. Here you actually have spending reduction equivalents. Then isocitrate is converted to citrate. Citrate is split to acetyl coenzyme A and oxaloacetate, and this is done with citrate lyase. And again, now we spend ATP, again from light reaction. So while TCA cycle, regular TCA cycle assumes production of reduction equivalents, and which is energy, here we have spending of reduction equivalent and energy, and where this energy and reduction equivalents come from light reaction of photosynthesis. Now, we have this acetyl coenzyme A, which is combining with carbon dioxide, so again capturing carbon dioxide, fixing carbon dioxide, and now we get pyruvate. Pyruvate is a result of this carbon dioxide fixation in combination with acetyl coenzyme O, and reactions is catalyzed with enzymes that are ferredoxin linked. Now we have pyruvate. That's more than enough. Pyruvate can be converted to phosphoenol pyruvate. Reaction spends ATP. Then PEP is converted to 3PG, later G3P and DHAP. So basically, like cycle of glycolysis just goes in reverse. Now we have two triosophosphates, G3P and DHAP, and those triosophosphates can combine to give hexosaphosphates or 6 carbon molecules like glucose, and that will be incorporated into cell material. So while in Calvin cycle you had three carbon compound called 3-PGA, here you will have classic G3P and DHAP. There are some key enzymes from this cycle, citrate lyase, so that's a key enzyme in reverse or reductive TCA cycle, and citrate lyase activity is used as a measure reductive TCA cycle in environment. So when you want to see the activity of this event, you will measure activity of key enzyme. So in mini summary, we have anoxygenic and oxygenic photosystems. Anoxygenic photosystem is linked to green and purple sulfur bacteria, heliobacteria, and green and purple non-sulfur bacteria. I gave details about green and purple non-sulfur bacteria in Word document. For the sake of time, I won't go through it. Oxygenic photosynthesis related to cyanobacteria and plants. Speaking about oxygenic photosynthesis, there are two photosystems, 1 and 2. In photosystem 2, which is the first player, reverse electron flow is used to make NADPH, and there is a quinone pool. In photosystem 1, there is ferredoxin and there is a menaquinone. Next one is acetogenesis. What does that mean? Creation of acetate. So acetogenesis can be broadly divided in reduction of carbon dioxide to carbonyl groups and to methyl groups. So let's go through steps of reduction carbon dioxide to carbonyl groups. Carbon dioxide gets reduced to carbon monoxide via enzyme carbon monoxide dehydrogenase. Now, carbon monoxide is attached to methyl group via acetyl coenzymal synthase. So two enzymes, dehydrogenase and synthase. The key molecule that is designed, created in this process is acetyl coenzyme A. And now acetyl coenzyme A is converted to acetate through Wood-Lungdale pathway, or since I really don't know how to pronounce it, I usually call it WL pathway. So this is ancient way of carbon fixation. Let's go through steps of reduction of carbon dioxide to methyl group. So this was carbonyl group, go to methyl group. So in glucose fermentation, we know that pyruvate is produced. The other product of glucose fermentation is hydrogen production. So pyruvate, hydrogen. Now, hydrogen is electron donor for acetogenic bacteria. And this is important to know. These bacteria that are creating acetate can use hydrogen as a fuel, as electron donor. Memorize that. Now, they also fix carbon dioxide. So hydrogen is oxidized via enzyme hydrogenase. CO2 is fixed. CO2 gets reduced. And then we have another cofactor that participates in this process, which is tetrahydrofolate. So in this reaction, tetrahydrofolate gets converted to formyl tetrahydrofolate. Next step is methyl tetrahydrofolate. So tetrahydrofolate is just a cofactor. Tetrahydrofolate has to be recycled because you need to have that factor recycled. And it's recycled with a purpose to join hydrogen and carbon dioxide at the beginning at reaction. Methyl groups are further used to create acetyl coenzyme A, and the enzyme that does this is synthase, acetyl coenzyme A synthase. And then the end product is conversion of acetyl coenzyme A to acetate through WL pathway. In summary, in acetogenesis, hydrolysis, so let's see, what's the first step? So we have breaking down or hydrolysis of some polymers, and that hydrolysis gives monomers. So you break down polysaccharides and you get sugars. If you break down lipids, you get fatty acids, or you break down proteins, you get amino acids. Now, those monomers get fermented. So it doesn't have to be only glucose. It can be any of those monomers. One of fermentation products is hydrogen. Hydrogen is a fuel. Hydrogen is oxidized carbon dioxide is fixed or reduced. Carbon dioxide is reduced to carbonyl and to methyl groups. The result of carbon fixation in both cases is acetyl coenzyme A. And then you have acetyl coenzyme A run through WL pathway, which gives acetate. Next process is methanogenesis. So very similar like acetogenesis. You have hydrolysis of polymers. You get monomers. Monomers get fermented to various products. One of products is hydrogen. Hydrogen gets oxidized as electron donor. Carbon dioxide is reduced or fixed. The result of carbon dioxide fixation is now methane and water, where in acetogenesis you had acetate. Here are some steps in methanogenesis. So in order to have this carbon dioxide reduced or fixed, you first have to activate it in methanogenesis. So the activation is done by methanofuran-containing enzymes. We'll call them shortly MFR. So once activated with MFR enzymes are reducing carbon dioxide and you get created formyl group. Formyl group gets transferred to methanopterin, MPT. So this is also cofactor, not tetrahydrofolate, MPT. The enzyme MPT reduces formyl group to methyl group. This is what you want. And methyl group is transferred to enzyme containing cobalt, COM. This is now methyl COM. Methyl COM is reduced to methane by reductase, and this is an enzymatic system. So you have methyl transferase and methyl reductase, but the whole purpose is to get a methane at the end. So the difference between methanogens and acetogens is that methanogens derive these methyl groups directly from methanogenesis, and acetogenesis creates this methyl group via tetrahydrofolate linked branch. And here is a brief summary of carbon dioxide fixing pathways. So there are purple sulfur bacteria where hydrogen sulfide is electron donor gets oxidized, and carbon dioxide is fixed via Calvin cycle. In green sulfur bacteria, slight difference, again, same electron donor, which is oxidized, and carbon dioxide is fixed via a reductive TCA cycle. Non-sulfur bacteria are different. So electron donors can be succinate or malate, any organic compound. They are oxidized, and carbon dioxide is fixed via 3-hydroxypropionate B cycle. This cycle is covered in your lectures in Word document. There are iron oxidizers. What's happening there is that iron, which is 2+ from pyrite, is what is oxidized, the source of electrons. Sulfate, SO4 2- can be electron acceptor, and result is iron sulfate and sulfuric acid. Fixation of carbon dioxide is done via Calvin cycle. Next one is ammonia oxidizing bacteria or nitrifiers, which we are going to cover later on. Here, ammonia is electron donor, and there can be various electron acceptors, oxygen, nitrite, and carbon dioxide is fixed via Calvin cycle. There are homoacetogens, where you should memorize that the electron donor in homoacetogens is hydrogen, and carbon dioxide is fixed via WL pathway. Acetate is final product. In methanogens, the same like in acetogens, donor is hydrogen, the same type of carbon fixation, WL pathway, and the final product is now methane and water. And I gave here a few clues about Rubisco. I think it's way more detailed in Word document. So this enzyme is specialized for carbon dioxide fixation and has two functions, carboxylase and oxygenase. What you need for fixation is carboxylase. You don't want Rubisco to be oxygenase. And there are those details in Word document that I uploaded. Here are some applications of this autotrophic or photosynthetic organisms that can make their own food. So since they are able to use light and fix carbon dioxide, pretty much always available source of food. So as the result of light and carbon dioxide fixation, microbes can be engineered to create specific products. So the fuel is there. We have carbon dioxide, there is a light, that's unlimited source. It's sustainable. So autotrophic microbes can be also used in microbial fuel cell, which is awesome. Like you can use hydrogen as electron donor and light to fix carbon dioxide. Pretty much, I would say cheap, but design is probably not so cheap and it's not so easy to do it. And again, autotrophy is characteristic of plants, and plants can be grown using light and carbon dioxide fixing mechanism. That's how you grow plant biomass. And then that plant biomass can be used as a carbon source for bacteria. And these bacteria can be engineered to make specific products. Again, the example of photosynthetic microbial fuel cell is given in details and in Word document. So this microbial fuel cell is using hydrogen as electron donor. Carbon dioxide is fixed via Calvin cycle or any other type of carbon fixation. Electrons are moved to anode. Again, they run through from one to another compartment, from anode to cathode, and they end up at various terminal electron acceptors. It can be oxygen created in hydrogen oxidizing carbon dioxide fixing photosynthetic processes. So basically, it's like a mini factory that is spending only hydrogen and the light, and that's it. I mean, it sounds great, which doesn't mean it's really easy to do. I have never done anything similar. I don't have any experience, but there are all sorts of literature available if you are interested. Homoacetogenic biofactory is another application of autotrophy. So again, hydrogen oxidation, cheap fuel. You have fixation via WL pathway. You get acetyl coenzyme A, and then you can create engineered microbe that can convert this acetyl coenzyme A to various pathways, and you can get various products depending on what you are interested in. And this would be the end of section of review session for Unit 8. And now my plan is to go to the section to the section 9, actually Unit 9. And I don't know what I pressed now. I hope I didn't mess up anything and I didn't stop recording, but I'm not sure. So let's go to the, I just want to check to see if we are still recording this. So give me just a few minutes. So let's see. I will now continue with Unit 9. And I hope you all can see this. Yeah, I'm not sure that yeah, okay. So this is it. Sorry about that. My technology skills are not the best. So Unit 9, microbiome. For me, this was easy unit, and it was interesting to listen what Lisa was saying about it, even though I'm myself not an expert for microbiome. There are some things from this area that I'm very interested in, which I will mention during the session. So I just gave a brief overview for you to be able to understand what is genetic, what is genomic, like a basic difference. So where genetic is the study of heredity, refers to function and composition of the single gene, genomic studies genes and their functions and refers to the function and composition of all genes and their internal relationships. So more like genomics is functional genetic. So the goal of genomics is to identify combined influence of genes on growth and development of one organism. And this is a little bit overview of history, which I don't think you will ever be asked, but it's really good to know for general knowledge who Craig Venter was, and that he is the father of modern genomics, and that he actually started this modern type of computer-linked science. So in order to spend months to sequence one genome, now you can sequence a genome like again, depending on the size of the genome, but for example, bacterial genome using those computer programs and modern sequencing, you can sequence in 1 day. One of those modern sequences is my favorite one, Illumina sequencing. And here is the YouTube link that I provided. I mean, maybe some of you actually saw how this looks. I recently learned about Illumina sequencing and I was impressed. So I encourage you to go to look this video. I mean, this is not for the exam, but I find it very interesting and cool to see how actually all sequencing process goes. It's informative. You understand much better what's going on. And again, this is the area of science that is rapidly progressing, and each year many new genomes are sequenced, like 64,000, as the literature says. The size of the genomes is different, of course. So bacteria that are live in soil, they have way larger genome than, for example, parasites, and there is a good environmental reason for that. So if bacteria lives in soil, bacteria deals with all sorts of influences and has to be adapted to many, maybe not so good environmental conditions. So they have to evolve genes to be able to adapt and to survive. On the other hand, parasites, they use many food resources and nutrients from the host, so they have smaller genomes. And I gave here example of first synthetic genome, Mycoplasma. So they created from Mycoplasma genitalium, they created Mycoplasma laboratorium. So their goal was to identify a minimal set of genes that are required to sustain life. And this is how they can this information they can use to rebuild a new organism. So they can use these crucial genes to synthetically create new organism. The bottom line is that nowadays you can synthesize organism in the lab. That's all what you need to know is which genes are minimal requirement for organism to function. So that's what those guys did. There are 482 genes in Mycoplasma genitalium. So they delete each of them individually, and as they delete one, they test whether this one can survive, and that's testing viability of those mutants. And then they found that there is a minimum of 382 genes needed to maintain life. This is important. Alkanivorax is important for you to understand. So for the exam, you need to know how we do microbial genomics and how we use this information in microbial physiology. And this example of Alkanivorax is important. So what we know from this organism, and again, this is just an example, you can take any other organism and do exactly the same. So this is gram-negative bacterium that grows on alkanes. It's detected in oil spills. So if we know that bacteria lives in alkane-rich conditions, that means that immediately tells us that bacteria has to have some genes to survive this stress, to be able, not just alkanes, but other stresses, because it lives in oil spills. It's not just alkanes that they use as the carbon source. There are also all sorts of other toxins, and there is a UV radiation that affects it. So bacteria has to, so it must be the genome includes genes that regulate stressful, that regulate metabolism under stressful conditions. So it has to be all sorts of adaptation to survive stress. Again, also genome has to contain genes for osmoregulations to survive oil toxicity, everything that I just mentioned, and also they have to be able to uptake carbon source that they have, which is alkanes. So they have to have alkane metabolism genes. So this is what we all need to know before we start researching this organism. What we will see is that when genes are close to the origin of replication, after we sequence a genome and we figure out where is the origin of replication, we know that if genes are close to this origin, they must be highly transcribed. And you know that if it lives on alkanes, probably alkane metabolism genes will be highly transcribed. So genes that are close to the origin of replication so they have to be accessed very easy by RNA polymerase. So that's why they're so close to origin of replication. If they are poorly transcribed, if they are far away from origin of replication, they're not highly transcribed. And now what we can find out from the example of this Alkanivorax borkumensis. So what we know, what kind of genes it has. It has permeases to be able to efficiently take nutrients from seawater. It has ABC transporters if they to be able to exclude some of toxins that enter in their organism. They have to be able to develop export system for all sorts of substances that are unwanted in the cell. They have also biofilm formation mechanism. Of course, since they live in salt in seawater, they have ion transporters for all sorts of transporters. Again, they don't have sugar transporters. They don't need it. They have transporters for alkanes. The main pathway is glyoxal shunt. There are no key enzymes of EMP and PPP pathways because, again, they don't eat sugars. They don't need to process sugars. And all anabolic reactions are relied on alkanes as the carbon source. So basically, when you see where the species live, you know what kind, you can not guess, but you can conclude which genes for which enzymes will be highly expressed. So when we talk about genome, we can study it and observe in 3 ways, as was suggested. Genome scale metabolic modeling, which is my personal area of interest, or GEM. We can do single cell genomic, which I never done, but I find it very useful, or core pan genomic. And we'll go through each of them. So GEM is a relatively new type of studies. So this is basically creation of metabolic map of an organism. And I love doing that because it gives you answers for so many questions. So what you do, you integrate all reactions in one organism observed from various levels. So since we have sequenced the genome, we can incorporate that genome into the computer simulation. So there are people who are doing this professionally, not myself, but we have collaborator who is creating us GEM for our methanotrophic organisms. So you integrate all reactions from genomic, transcriptomic, proteomic, metabolomic. When you incorporate all those information, GEM is created, and that's computational representation of all reactions in organism, which means you can simulate, for example, deletion of one gene or addition of gene. And you can predict how organism will behave. So let's say I want my all carbon from methane to go to production of specific compound. And I don't know where my carbon is spent. I don't know where is the bottleneck. I can guess, I can conclude, but in order to see is that true, I will have to go through all experimental setup to delete a gene and then to test whether my hypothesis was correct. With GEM, you can do this like it can be just computer based. You delete it in program and you click a button and it tells you where your carbon is going. So you know whether there is any purpose to do that deletion or you should look somewhere else. So it's pretty cool how you can do that. Again, it's not really all so easy as it seems, but I just said in the most simple way. Single cell genomic is the type of genome analysis that helps to discover how mutations are selected and how that influences evolution of organisms, because some mutations are useful. So you can see in cell, on the single cell level, how mutation is developing. Core pan genomics, so just to understand what is core, what is pan genome. So know that one species has various strains, and the core genome represents a set of genes that is shared between all strains, so they all have some same genes, but pan genome includes those individual genes, individual sequences that originates from phages, from transposons that are different, and pan genome is actually separating strains. So even though it's the same species, there are different strains. So metagenomic is very popular these days, and basically the way how that is explained is in one environment you have all set of DNA sequences, and all these sequences are called metagenomic sequencing. So you are analyzing DNA from one environment, which helps you to build DNA map, and this is a step, this is used in microbiome. So once when you have the DNA map, you know which sequences exist in, like, let's say, gut or in soil or in any other environment. This is a basic step. This is the basic step that needs to be used later. So, and that's used by microbial physiologists to understand the map. So you need to be, it's not only important to have DNA map, you need to have microbial physiology and biochemistry knowledge to be able to understand that map. Solid DNA map, metagenomic, means nothing. So this was a basic, as I said, for developing of microbiome. So basically what is done to generate human microbiome is collecting a DNA sequences from human gut. Then you identify members of these communities, and then you study metabolic potential of these species. Again, you need to know microbial physiology and biochemistry. And here is how we study microbiome. So basically, people are fed with a different specific foods, then you collect fecal samples, analyze those samples, and you determine changes if there are any. And usually there are. So again, what we can do with microbiome is, like, there are so many things that you can find out. You can observe how metabolic responses in detected microbes are changing depending on the different diet. You can compare how microbes from similar in different environments. You can compare microbiomes from people that originate from different geographic locations, and you can basically see how much we are eating bad food comparing to, let's say, Eastern cuisine. So this Western orientated food, Western food apparently is not good. So I recently learned that we eat unhealthy, and I never considered that. I like food that I eat, and I don't think I would be able to live on healthy food only. But again, that's how we are used to. Maybe we wouldn't be even well adapted to Eastern cuisine. I don't know. And apparently there are no clear evidences that causes big problems. I mean, microbiome is changed definitely, but it doesn't necessarily mean it causes some harmful effect. Or maybe there are some studies that are proving, again, I don't know too many things about that. I would never study microbiome, not very interested in that. But what you can do, you can simulate controlled environment, and that's done by using germ-free animals, and then you feed them with different food, and you follow how microbes are developing in their gut based on food. Good thing is that apparently we can change our microbiome by just changing our nutrition, which is good. If one day I really want to be healthy, I will start eating normal food. I don't think that will happen soon. So this is the end of session 9, and I won't make a break now. I will just go straight forward to the unit 10, since I think we are already short with the time, but doesn't matter because I'm anyway converting this to YouTube video. So unit 10, very important for this exam, sulfur nitrogen. Personally, I love sulfur cycle. For me, that looks easy. It's easy to memorize, but again, maybe it's just my personal preference. So there are 4 forms of sulfur: sulfur hydryl, sulfides, elemental sulfur, and sulfates and sulfides. We can find sulfur in sulfur minerals and sulfide minerals, gypsum or pyrite. You all heard about this. So when we talk about sulfur cycle, we will start with, so there are sulfur oxidation and sulfur reduction. So we will start with oxidation as the first process. So in order to begin oxidation process, cell needs to have some type of reduced form of sulfur. That's a precondition. And reduced form of sulfur is hydrogen sulfide. You've been seeing this in anoxygenic photosynthesis. And this is the first source of sulfide, anoxygenic photosynthesis, green and purple sulfur bacteria. Another sources of this reduced form of sulfur are elemental sulfur, just it has to be reduced to get hydrogen sulfide. Also, thiosulfate can be reduced, and then you get hydrogen sulfide. Basically, reduced form of sulfates are giving you H2S or hydrogen sulfide. And also, when there is desulfurylation of organic compounds, when organic compounds decompose, you can get hydrogen sulfide, which is a starting point electron donor for sulfur oxidation. And here is all process of sulfur oxidation. So hydrogen sulfide is oxidized to elemental sulfur. Electrons from this oxidation travel to flavoprotein, and from flavoprotein, those electrons go to typical quinone pool. Now you have reduced quinone H2, and these electrons from quinone pool are transferred to cytochromes B and C. This is the place where proton motive force is formed. So electrons are transferred from quinone to cytochromes, and protons are transferred across membrane to generate proton motive force. From cytochromes B and C, electrons travel to cytochrome AA3, and terminal oxidase is oxidizing cytochrome and reduces some terminal electron acceptor, which can be oxygen. Again, at the terminal oxidase, you also have proton motive force formed. So it's quinone pool and terminal oxidase. The final product, one of products, is elementary sulfur, but that's not the end. Elementary sulfur can be further oxidized, and electrons are released and travel again to cytochromes to join electron transport chain. So all purpose of oxidation is to get electrons that will be transferred to cytochromes and later to terminal oxidase. Just as a result of sulfur oxidation, we get sulfide, SO3, and SO3 is oxidized to SO4, 2-, which is sulfate. And this is done by one simple enzyme. Also, sulfite can be converted to sulfite via a more complicated enzyme, which is called APS reductase. No, but those details are given in Word document. Also, sulfur oxidation can be related to sulfur crystals. So very often elementary sulfur is in the form of crystals. Microbes simply attach to those crystals. Then they oxidize elementary sulfur through sulfite and getting sulfate, SO4. And this oxidation from elementary sulfur to sulfate is releasing electrons. Those electrons usually acidify the soil where this process is happening. And very often this strategy of microbes that can oxidize sulfur crystals are used to acidify alkaline soil. Sulfur oxidizers can be used in bioremediation, and here is how. Like when we have oil and gas refined that generates elementary sulfur, you will guess elementary sulfur can be used as electron donor, because if you saw, as Lisa mentioned, those pyramids where elementary sulfur is piling up, this is like a food source, electron donors. So they oxidize elementary sulfur. That's how they detoxify this sulfur. And what they can also do, they fix carbon dioxide, and they can detoxify sulfur waste. Sulfur oxidation is widely seen in marine ecosystems, and it's for me this is very cool animal that I found out recently. It's really interesting how symbiotic relationships are developed between microbes and all sorts of different organisms, especially in the sea. So this is symbiosis between some marine animals and sulfur oxidizers. So here's what's going on. So these animals have an organ called plum, and that's where they receive gases needed for all other processes. Gases are hydrogen sulfide, so electron donor, oxygen, electron acceptor, and carbon dioxide, which will be fixed. So this is all in plum. In other organ, trophosome, there are bacteria, sulfur oxidizers, that will use everything what plum picked up from the environment. So they have electron donor, hydrogen sulfide. They have oxygen picked up again in plum, so that's electron acceptor. And the sulfur oxidizer also fix carbon dioxide, and they create organic fuels for the animal. So animal picks a food source for microbes, and microbes are giving them another food source. So it's like exchange. Really cool. The next step in sulfur metabolism, the next part of sulfur metabolism is sulfur reduction. And so this all till now was oxidation. Now we go to reduction. So all process of sulfur reduction starts with ATP in cytoplasm. So there is an enzyme, ATP sulfurylase, that cleaves PPI, pyrophosphate from ATP, and you have AMP left. Now, in next step, ATP sulfurylase adds sulfate to this created AMP, and the resulting compound is APS. So this APS has two branches. The first one is assimilative metabolism, and second one is dissimilative. So let's go first through assimilative metabolism. So when you have AMP connected to sulfate group and APS as a starting component, there is an enzyme kinase that adds one more phosphate group, and you get molecule PAPS. Now it has two phosphate groups. PAPS is further reduced to sulfite, and sulfite is reduced to hydrogen sulfide. Hydrogen sulfide is shunned further to assimilative metabolism to create organic sulfur compounds like amino acid methionine or cysteine. And if we have dissimilative metabolism, everything is the same up to formation of APS. But now, instead of APS kinase that forms PEPs, you have APS reductase that creates sulfite. Sulfite reductase creates hydrogen sulfide. So pretty much it's the same, just like in assimilative, but the fate of hydrogen sulfide is different. In previous case, in assimilative one, let's go back, H2OS, hydrogen sulfide, goes to metabolism to be used for assimilation, but in dissimilative metabolism, hydrogen sulfide is excreted. Now, how is proton motive force generated in sulfur reduction? So what you have is electron donor hydrogenase that oxidizes hydrogen, and electrons travel to cytochrome C3. And now from cytochrome C3, electrons go through another membrane spanning protein, which is typical for this process, HMC. And HMC has ability to deliver electrons and reduce sulfate to sulfate. Sulfate can combine with ATP to create APS, and then APS is used for dissimilative metabolism to generate hydrogen sulfide. Some characteristics of sulfur reducers, which you could probably conclude, is that they have a very short electron transport chain, and sulfate is not a very good electron donor, should be electron acceptor. Sulfur is very poor electron acceptor comparing to nitrate and oxygen. And electron donors for sulfate reduction have to be more electronegative compounds. So this is a mistake. So we talk about sulfate as electron acceptor. The same how oxygen and nitrate are electron acceptor, just they're much better. Donors are different. So donors are oxidized, so we can have electron release. So there are no many choices for sulfate reducers. They can oxidize either organic compounds or hydrogen because they are more electronegative than electron acceptor. So sulfate reducers are usually in competition with homoacetogens and metanogens, and you can conclude why. Because in order to get sulfate reduced, or they have to oxidize some electron donor that are more so they have to have good electron redox couple, and that can be only hydrogen or organic compound. So if there is no organic compound, they have to take hydrogen, and hydrogen is source of electrons for acetogens and metanogens, so they are in competition. When hydrogen is oxidized or organic compound is oxidized and sulfate is reduced, as a result, we get hydrogen sulfide. And now hydrogen sulfide is an electron donor for sulfate oxidizers, which we covered first. And here are some interesting things about sulfate reduction. So how corrosion forms. So there are those microbes that will consume oxygen and some metal surface, and as they're consuming oxygen, the environment becomes oxygen depleted. And this is a great environment for sulfur reducing bacteria, or SRB. So they reduce sulfate. So in this case, sulfate is electron acceptor, and they create hydrogen sulfide. And when hydrogen sulfide reacts with metals, that forms metal sulfides, which create corrosion. And here is another interesting example of syntropic interaction, which I personally really like because I like methane oxidation. So in anoxic environment, methane can be also consumed, which is weird because methane oxidation usually requires oxygen. But those organisms that are oxidizing methane without oxygen are not bacteria, they are methane oxidizing archaea. And so methane is electron donor for them. So they're oxidizing it even without oxygen, but as an electron acceptor, they have sulfate. So this is basically called reverse methanogenesis. So methanogenesis is a process where you make methane. This is reverse, so standing methane. So methane is oxidized and sulfur is reduced. Again, sulfur cycle is connected to other processes such as pyrite formation. Sulfur oxidation is usually linked into denitrification, and we have organic sulfur cycle. So pyrite formation is probably, we are very familiar with it. Again, sulfur reducing bacteria are creating sulfide, and sulfide can interact with iron minerals. That's how corrosion forms, as we mentioned earlier. Resulting compound, iron sulfate interacts with elementary sulfur, and you get iron sulfide, which is pyrite, sorry. Sulfur oxidation linked to denitrification. In this case, you have hydrogen sulfide as electron donor, where terminal electron acceptor is nitrite, not oxygen. Nitrate and oxygen are both good electron acceptors. Of course, oxygen is better. And as about organic sulfur compounds, very interesting process where you have a molecule made by marine algae, DMSP, and microorganisms can degrade this DMSP, then newly created DMS, so it's basically removing a phosphate. DMS is produced in ocean, and that's now electron donor. Electron donor in terms of, so DMS is electron donor, but it's photo-oxidized. And this photo-oxidation of DMS is creating some aerosols with sulfides and sulfur dioxide. And these compounds are contributing to global cooling. So this process is used in geoengineering. And now let's go to nitrogen cycle. We already mentioned some parts of nitrogen cycle. And this is something that you do want to know basically in details. So all process of nitrogen cycle starts with fixation of atmospheric nitrogen. We already talked about nitrogen fixation by nitrogenase enzyme, oxygen sensitive enzyme. So I'm not going to go again in details about that. But what you need to know is that nitrogen fixation is really unique for bacteria, and it's completed by nitrogen fixing bacteria that contain that oxygen sensitive enzyme nitrogenase. Again, whole point of nitrogen fixation is to get biologically available form of nitrogen, which is ammonia, NH3. Now, ammonia is source of energy for organisms ammonia oxidizing bacteria, or AOBs. So these organisms are oxidizing ammonia, so ammonia is electron donor. As the consequence of ammonia oxidation, these bacteria create nitrite, NO2 minus. Now, nitrite is a substrate for specialized bacteria called nitrite oxidizers. As the name says, they oxidize nitrite to nitrite. So NO2 minus to NO3 minus, that's oxidation. Nitrite, NO3 minus, is substrate for organisms called denitrifiers. They reduce now NO3 minus to NO2 minus, and NO2 minus is also reduced to dinitrogen. This product, dinitrogen, is now again fixed by nitrogen fixing bacteria, and the cycle goes on. So there are different perspectives of nitrogen cycle, and I presented 3 here. And I strongly recommend you to read the review paper that Dr. Stein posted online, and to look picture of nitrogen cycle and to try to memorize what's going on. I personally like seeing picture, and it helps me memorizing process. So from this perspective one, we have start of nitrogen, we will start with biologically available form of nitrogen, which is ammonia. Ammonia oxidizing bacteria, along with nitrite NO2 minus oxidizers, are completing so-called nitrification. So these are two groups of organisms, ammonia and nitrite oxidizers. But the final result of these two processes, which is these two processes are together called nitrification, we get creation of nitrite NO3 minus from ammonia. NO3 minus now goes through assimilative nitrite reduction. So it has to be reconverted to ammonia. So when converted to ammonia by specific enzymes, it is converted into amino acids and nucleic acids. So, for example, if methanotroph is fed with nitrite, NO3 minus as a nitrogen source, they have to reconvert it to ammonia with assimilative nitrate reduction to be able to use that in their biosynthetic processes. If you feed methanotroph with ammonia, usually it would be expected to have less energy spent, but that's not the case for other reasons, and I'm not going to go in details now because it's not part of the class. In the process of ammonification, which is assimilative nitrate reduction, organic matter is going through decomposition. So when ammonia is incorporated into amino acid nucleic acid. So now we have biomass built. So in next step, this biomass can go through the process of ammonification. So organic matter is going through decomposition and mineralization. And now this ammonia from biomass returns into the cycle. So it's first made through assimilative nitrate reduction, goes to the biomass. Biomass is now decomposing, and you have return of ammonia, which is a substrate for ammonia oxidizers and NO2 minus oxidizers. Nitrogen cycle from perspective 2. Again, we start with ammonia as biologically available form of nitrogen. And here we have organism that has it all. So if you remember, we had in previous one, we have ammonia oxidizers and NO2 minus oxidizers, which are completing nitrification, conversion of ammonia to NO3 minus nitrite. These COMAMOX are doing everything together. So they're completing fully conversion of NH3 to NO3 minus. Then NO3 minus goes to assimilative nitrite reduction and is converted to NO3 minus. And again, incorporated into amino acids and nucleic acid, organic matter decomposes, and ammonia is returned into the cycle. Next one, NMOX. What does that mean? So these are bacteria that are completing nitrogen cycle in the way how I call it, perspective 3. So they're very special. They are utilizing ionic form of ammonia or ammonium, which is ion, and they combine it with nitrite, NO2 minus. This reaction creates nitrogen, dinitrogen, and then nitrogen fixing bacteria with nitrogenase are fixing nitrogen, and they create biological available form of nitrogen, which is ammonia. Ammonia is then source for ammonia oxidizing bacteria. So they are oxidizing it to nitrite, and then nitrite is a substrate for NO2 minus oxidizers, and you get NO3 minus. Then NO3 minus is a substrate for denitrifiers. So these organisms, denitrifiers, are reducing NO3 minus to NO2 minus, and then further to NO, and then N2O, and sometimes to atmospheric nitrogen. And then atmospheric nitrogen can be further fixed by nitrogen fixing bacteria, and cycle continues. So roughly, nitrogen cycle can be divided in 3 groups: nitrification, denitrification, and NMOX. So nitrification is aerobic. So this is like a mini summary. Nitrification is aerobic part of nitrogen cycle. And you have ammonia oxidation completed by AOBs. Example is Nitrosomonas europaea. These organisms are chemolithotrophs, so they oxidize ammonia, inorganic compound, to get an energy. They oxidize it NH3 to NO2 minus. They also fix carbon dioxide. So these bacteria are called ammonia oxidizing bacteria. Here is how all process of ammonia to nitrite oxidation goes. So it has few steps. So first, ammonia monooxygenase is oxidizing ammonia to hydroxylamine. Then hydroxylamine is oxidized via dehydrogenase to nitric oxide, and nitric oxide is converted to NO2 minus nitrite via unknown enzyme. This ammonia oxidation also has some electron transport chain, to generate proton motive force. So here is how that goes, slightly complicated. I'm also learning about that on daily basis. So monooxygenase requires electrons to activate oxygen. So those electrons are coming from quinone pool. When electrons are delivered, molecular oxygen is activated. Now, one atom from that molecular oxygen is then attached to ammonia, and we get hydroxylamine. Next, hydroxylamine is oxidized by dehydrogenase. One portion of electrons from this reaction goes to quinone pool, and second portion from this reaction goes to ammonia monooxygenase to continue activating oxygen so the cycle can operate. From quinone pool, electrons travel to cytochromes C, to cytochromes, and from cytochromes, electrons travel to terminal oxidase. At terminal oxidase, there is a reduction of oxygen to hydrogen dioxide. And again, quinone pool and terminal oxidases are places where proton motive force is formed. In part 2, so now we have created from ammonia to NO2 minus. Part 2 is completed by NO2 minus oxidizing bacteria. They have to live in close association with ammonia oxidizers because ammonia oxidizers create a substrate for them. So then nitrite is oxidized, and here is how electron transport chain looks. So basically enzymes, NXR, there are all sorts of enzymes that are oxidizing NO2 minus to NO3 minus, nitrite to nitrate. Electrons from this oxidation again go to cytochrome, and from cytochrome C, they go to terminal oxidase. At terminal oxidase, there is reduction of oxygen to water, but here there is no quinone pool. PMF is only created at terminal oxidase, and most of this PMF goes to reverse electron flow because they need to make NAD needed for carbon dioxide fixation. And now there is a bacteria that does it all, Comamox. So again, ammonia oxidizers and NO2 minus oxidizers are completing this whole process of nitrification. So first group is oxidizing ammonia, second group is oxidizing NO2 minus. So Comamox are doing it all. So they complete all nitrification process by themselves. So from ammonia to NO2 minus. And the model organism is Nitrospiram. So they're also ammonia oxidizers, but way more efficient than regular ammonia oxidizers. So here is the example how we can use this Comamox how we can apply this, and that's done in Dr. Stein lab. So we have those, not we, Payment does it. They have those fish tanks where fish are growing and they're fed with all sorts of fish food. So as they are metabolizing food, they excrete ammonia, NH3, and that ammonia created by fish can go to microbial bioreactor. And bioreactor is fulfilled with efficient ammonia oxidizers, Comamox, so they complete full ammonia oxidation, and they end up with NO3 minus production. This is nitrite enriched water, so NO3 minus water, and this water can go to feed agricultural plants like a fertilizer. So all what we talked about, nitrification, was aerobic part of nitrogen cycle, where denitrification is anaerobic, so minus oxygen. So denitrification is dissimilatory respiration of nitrogen oxides, and it can be observed as nitrate respiration. So the same how bacteria breathe oxygen, they can breathe nitrite, NO3 minus. it can be observed as respiratory denitrification and eukaryotic denitrification. So how nitrate respiration functions? So this is dissimilatory nitrate reduction. So nitrite NO3 minus can be transported through some methods, membrane or soluble transporters. So it's like imports through membrane. In the same time, NO2 minus is exported from the cell. So I would say this is an antiport. It can be also, I don't exactly know. I think there are various forms. NO3 minus gets reduced to NO2 minus, and NO2 minus is exported out. Now, what's happening is next. NO2 minus is electron acceptor. So bacteria are now not using oxygen. They're using NO3 minus as terminal electron acceptor when there is no oxygen. There are some changes that are happening when we switch from oxygen to nitrate. So instead of quinone, we have menaquinone. Bacteria loses cytochrome B, and there is no terminal oxidase, there is terminal nitrite, NO3 minus reductase. So this is nitrate respiration that results in NO2 minus production. NO2 minus is toxic. So basically, each organism that breathes NO3 minus has to have some detox or system or sink for NO2 minus because it's toxic. So in respiratory denitrification, process is similar, but slightly different. So NO3 minus again gets reduced to NO2 minus. So when you had a nitrite respiration, NO3 minus reduction to NO2 is the end, and then NO2 minus goes somewhere to be detoxified. In respiratory denitrification, process is longer, so you have various enzymes that are, of course, reducing now this NO2 minus to NO, which is nitrogen oxide, nitric oxide. Then NO gets reduced to nitrous oxide, N2O, which is greenhouse gas, very harmful. And some organisms can also reduce NO2, N2O, nitrous oxide, to molecular nitrogen, dinitrogen. So sometimes ends up at nitrous oxide, sometimes goes further to get atmospheric nitrogen. So in denitrifiers, there is a specific electron transport chain. So they can consume organic molecules and produce NADH. So electron donor can be any organic compound like glucose. NADH is beginning of electron transport chain. It gets oxidized and you have electrons transferred to series of quinones and cytochromes. To terminate the electron transport chain, electrons are delivered to nitrate reductase. So there is no terminal oxidase. And NO3 minus, which is electron acceptor, is reduced to NO2 minus. Another electrons are coming in to continue reduction from NO2 minus all up to N2O or N2. And this process contains a few, actually all sort of denitrifying enzymes. For NO3 minus, for NO2 minus reduction near K, near S. So what this enzyme is doing is reducing NO2 minus to NO. Then nitric oxide or NO reductase, which is reducing NO to nitrous oxide N2O. And some organisms have nitrous oxide reductase, which is reducing N2O nitrous oxide to dinitrogen. And last but not least are eukaryotic denitrifiers. Example one is Fusarium, it's a fungi, and second one is foraminifer. So in mitochondria, in Fusarium, there are enzymes nitrite and nitrite reductase, and when there is hypoxia, they are starting denitrifying pathway. In the cell, there is all sorts of pyruvate, and pyruvate is oxidized to acetyl coenzyme A. They specifically convert acetyl coenzyme A to formate, and formate get oxidized, formate gets further oxidized, and electrons from these oxidations are used to reduce NO3 minus to N2O nitrous oxide. So through all NO3 minus, NO3 minus, NO2 minus, then NO, N2O, which is the final product. Simultaneously, they still breathe oxygen and produce H2O. This is high, so they do hybrid respiration. They do this in conditions when there is a lack of oxygen. In foraminifers, so it's a specific type of denitrification, and this organism actually does complete denitrification to molecular nitrogen. So fusarium goes to nitrous oxide, foraminifer goes to dinitrogen. They usually live in an anoxic area, of course. They don't have oxygen, but they still grow. And the way how they grow is that they accumulate NO3 minus inside the cell. And when that labeling experiment was done, it was concluded that all imported nitrite NO3 was converted to dinitrogen. So they definitely do denitrification. Special organisms, anamocs, are known as, we already mentioned them, but little in details. They're doing anaerobic ammonium oxidation. So for them, substrate is not ammonia, it's ammonium, so ionic form. And in the same time, when they oxidize ammonium, they reduce NO2 minus. And as a result, we get dinitrogen and water. They would be really useful in wastewater treatment because they can efficiently remove ammonium from these anoxic areas. They have very specific organelles, so it's a bacterium with organelles. Those organelles are called anamoxisms, and you need to be aware and to know what is the function of anamoxisms. So these are membrane-bound structures, and they are placed for ATP production. And here is how anamoxism works. So they fix carbon dioxide via acetyl coenzyme A pathway. NO2 minus provides electrons to reduce carbon dioxide to methyl group. Now, when we have methyl group and carbonyl group, this probably sounds familiar, there is a complex enzyme dehydrogenase that connects these two groups, methyl and carbonyl, together. The result is acetate. Acetate converted to acetyl coenzyme A, and acetyl coenzyme A is metabolic precursor used for various biosynthesis. That's all happening in anamoxisms. So the source, so anamoxisms have to have, anamox has to have nitrate, NO2 minus. So what is the source? It's aerobic ammonia oxidation by AOBs, also nitrate reduction. And those anamoxisms fill up all cell of anamox, and they're like a special component surrounded by lipids. They're placed for ATP synthesis. So they have specific lipids, and know these things. These lateral lipids are protection from some toxic intermediates that are produced during processes in anamoxisms. Hydrazine is one of them. And anamox has specific ETC. Here is how it goes. So ammonia is co-substrate with ammonium with hydroxylamine. So that originates from ammonia oxidizing bacteria. Hydrazine hydrolase creates hydrazine, and hydrazine oxidizing enzymes will complete hydrazine oxidation, which will create proton motive force. So proton motive force originates from hydrazine. Electrons from this oxidation are used for NO2 minus reduction. So it's like a reverse hydroxylamine dehydrogenase, so not the one that we know from ammonia oxidizers. And this is the end of this session. Again, I will convert this to YouTube video and post it online, and see you tomorrow when we are going through actual exam. So I hope to see you all later tomorrow.